about this stuff now. So let me pull up let me do my share screen thing. I got to get to, where is my, okay. Oh, for goodness sakes. Okay, there we go. Let me share my screen with you. That one, I'm going to share that. And let me get to, where is it? Oh, come on now. There we go. All right, can you see this? The PowerPoint. Is that a yes?
Yep.
Okay, good. Because I can only see 1, 2, 3, 4, 5 people at a time. That's it. So never know. I can see Anastasia's ceiling right now, so I'm assuming there's a live body there. Oh, there she is. Okay. All right. So what I'm doing is I'm jumping over the liver. So in your books, where we are is over on page 902. Okay. So, and by the way, if you can stay on after this, I want to talk about some lab stuff and pick your brains. I'm hoping y'all have looked in there because most of you are in my lab one day or the other. Okay, so small intestines, this is where we're going in to really do most of the digestion, certainly most of the chemical digestion, and the mechanical digestion, the segmentation is going on in there. So the big thing for the small intestines is this is where most of our nutrient absorption occurs, our amino acids, our lipids, our nucleic acids, the vitamins, electrolytes, actually even most of the water. It's all absorbed in this most proximal portion in the duodenum and the jejunum, because this is the portion where it's kind of our body has a philosophy of let's get it done and grab it up while we can. Then the rest of it, as you're moving along through the track, it's just about kind of grabbing up the remains, okay? So it's a big deal if you lose your small intestines. The ileum, its job really is, its primary job is reabsorption of bile salts. And that is a big deal because, you know, your liver is making this bile as the detergent. So it needs to recycle it so that it can keep making more bile so that you can keep breaking down the fats. Bile isn't an enzyme, though, of course, it's a detergent. It's a very different thing. It's a cholesterol thing. Okay, so when you look at the small intestines, one of the big things is all these extra folds, okay? So the surface area is huge. You've got plicae circularis, which are the circular folds. Actually, that's what it literally means. They're not rugae. They are permanent transverse folds. They don't stretch out. They're just there. Then you also have on those folds, you've got your villi, those things sticking out. And then on the villi, the simple columnar has microvilli. So you've got fuzz on your fingers, on your folds. It's like the ultimate plush terry cloth towel for absorbing stuff. It's surface area galore, all right? The mesentery proper is what's holding all this stuff. So remember, that's that visceral peritoneum that's coming together. And that's where you've got all your blood vessels, lymphatics, nerves, all that good stuff. And we also like to store fat. One of the things about mesenteries is that our body says we don't know when our next meal will come from. And now, of course, we're not sure when we can go to the grocery store. But it is the place where we stash fat just in case, because biologically speaking, we're prepared for the big starvation. It just hopefully doesn't seem to come now. All right. You need to know the layers are just the same. We've gone through this where we've got the inner circular, outer longitudinal of those muscles. Then we've got the mucosa, and the big deal is it's all about absorption. It's got scattered goblet cells. So we do produce mucus to kind of help things move along, but the biggest thing is that surface area for absorption. The different parts of this are the duodenum, jejunum, and ileum. They get longer and longer as we go along. Duodenum's only about 10 inches long. So let's see this picture. So it just wraps around the head of the pancreas. It is actually retroperitoneal. It's the place where we have the joining of the common bile duct as well as the main pancreatic duct at this hepatopancreatic ampulla. It lets into the duodenum through this little duodenal papilla. We have actually, there's an extra just accessory duct or duct of Santorini that's kind of draining constantly from the pancreas to let in some bicarbonate to make sure that we don't burn out our duodenum with that acidic chyme coming from the stomach. But anyway, so we've met this in the lab. Has everybody seen this in the lab? Remember, I know it's been a few weeks at this point, but does that look familiar? We good? This was with the pancreas on a stick. And we also had the whiteboard, you know, the expensive one. We had one that was broken and the other one that was intact. So they showed this really well. So you'll need to know about that major duodenal papilla poking into the duodenum to release all the pancreatic juice and the bile so that when we eat something, this can deal with every aspect of every nutrient that comes in, okay? It's controlled by the sphincter of Oddi as to when we release things. We'll get to that in a bit, all right? After the duodenum comes the jejunum, which then leads to the ileum. The jejunum is probably about 8 feet long. The ileum's the longest. It's about 11 feet long. All right, so those are kind of the big things. The ileum we had met in the lab because the ileum has those little gut-associated lymphatic tissues that we know as the Peyer's patches, right? So we met those, the little lymphatic nodules. And we met the lacteals. Remember what goes into the lacteals, anybody?
Dense capillary bed. And
well, what goes into the lacteals? What do we absorb into them, remember?
Fat.
Fat, that's right. So chyle is the fat stuff into the lacteals. All right, cool beans. All right, at the end of the ileum is the ileocecal valve, and then we get into the cecum. So we'll worry about that in a minute. All right, let's see. Other things that are new. So we've talked about microvilli. The other name for them is brush border. So when we look at the final clipping of the nutrients we eat, we keep chopping from big chunks into smaller chunks, into smaller chunks, into smaller chunks until we get to monomer units. So back in the chemistry chapter, chapter 2, we had the remember, the basic building blocks of carbohydrates were monosaccharides, right? Glucose, fructose, galactose, these things. And then they build those into disaccharides like sucrose, maltose, lactose, those things. And we keep building them up into the oligosaccharides, into the polysaccharides. We got starch, right? So the microvilli, the brush border, makes enzymes. These simple columnar epithelial cells actually will produce the enzymes that will do the final clipping. So they'll do something where they have like lactase. You know how some people can't eat lactose containing things, the milk sugar? They're lactose intolerant. That lactase is made by these small intestinal cells and released at the brush border. So they're called brush border enzymes. We good so far?
So the microvilli are brush border enzymes?
They're called brush border enzymes because like these little dipeptidases, disaccaridases, like lactose is broken down by lactase. These are brush border enzymes. It's a big category, just says they are made by the small intestinal cells and released at the microvilli. Better?
Yes, thank you.
Okay. All right, let me let Denise in. She's found her way back. So Denise, you should be here now. Woohoo! I hope. Okay, so anyway, let's see what else. We've got crypts of Leverhulme. I just like saying Leverhulme too. The intestinal crypts, that's these guys, okay? So the crypts of Leverhulme, the intestinal crypts make intestinal juice. It's kind of this slightly alkaline, watery, mucousy stuff that it's a great carrier for the nutrients that come in that we're absorbing from the chyme. Okay, so it's just part of that solution thing, and it helps neutralize the chyme a bit. Paneth cells that you see over here on the lower right, their job is to make our most common thing for antibacterial. Remember lysozyme? Our body's philosophy is, if you're going to the surface, take an antibacterial. So the Paneth cells make the lysozyme. The other thing in here is, just as with the stomach, there are a whole lot of stem cells. Stem cells down at the bases of the crypts. These guys are going to make more of whatever it is that we need, just as in the stomach, okay? Again, this is why chemo that attacks anything that is rapidly mitotic, that's why it has such a terrible effect on the GI tract, causes nausea with the stomach, causes diarrhea with the intestines, okay? So that's the why. Everybody good with those? Okay, well, down in the base of the duodenum, we also have duodenal glands or Brunner's glands. They make an alkaline mucus. Their job, therefore, is to help with neutralizing that nasty acid chyme, because we don't want the duodenal mucosa to be ulcerated. Okay? All righty. So far, so good. All right. So I love this picture. They don't really look like a glowing pink thing, but I like the way they stained this and lit it up. All right, so everybody should have seen this wonderful thing when you were doing your assignment for the homework, for those who did it, because this was like the perfect, helpful thing. If you didn't utilize this, you weren't paying attention to your book, right? Just saying. Okay, so anyway, I would say that when you want to make sure that you've got the basics of this system, on page 904, it is key because it talks about everything that's going on and it does it in order. It matches everything up for you, okay? So tell me, with neural control of all this, parasympathetic, what does it do? Stimulate or inhibit?
Stimulate.
Yes, it does. So what does the sympathetic do?
It inhibits.
Inhibits everything.
It inhibits. Okay, so well then how is it that if you scare somebody really badly, they might poop their pants?
Okay.
Aren't you on sympathetic mode when you get scared?
It
Wanna
is
know why?
transition from sympathetic to parasympathetic.
Nope.
Maybe
That would be,
relaxing your sphincters.
you forget to control your sphincters. That's all it is, right? You already made it. Your GI tract has slowed down all of its activity, but you already made the feces, so it was sitting there in your rectum, and then when they scared you badly enough, you totally forgot to hold on to it. That's all it is, okay? Same with the urinary system. We can scare the urine out of you too, right? You ever heard the expression, right? Oh my God, they scared the pee out of me. All right. So anyway, there's a lot of digestive stuff that's going on in the small intestine. So let's move on down to the small intestine over here. Okay, so in the small intestine, there's mechanical. We know about that because we've done the segmentation, already looked at that. We've got chemical digestion. So the bile that's coming is just helping break down the fats, but the chemical digestion is all the enzymes that are coming from the pancreas. They're the brush border enzymes being made by the small intestines themselves. Okay, when we're doing all of that digestion, then we're ending up with our monomer units. So we're doing a lot of absorption. Small intestine's really important. There are some kids who are born with what we call short gut syndrome. When they don't have a small intestine, they're not going to be able to absorb food from their diet. They will have to get their nutrition through another way, through their blood vessels. So I'm really grateful to have my small intestines because I think eating stuff is fun. So there you go. All right. So anyway, in order for your small intestines to do their job, the liver has got to be able to deliver bile. We're going to talk about the liver shortly. We've got to have bicarbonate ions from the pancreas because we've got to neutralize the acid chyme. We've got to have all the digestive enzymes from the pancreas because otherwise you're not going to be able to digest what you need. And we've got to have a really slow delivery of the chyme. So remember when we talked about the stomach, right? That pyloric sphincter, we had this whole enterohepatic and enterogastric reflex going on, because we need to make sure that we are not going to bolus through too much, right? So we make that pyloric sphincter of ours control forward flow of the chyme into the duodenum. We don't want to burn it out. So we've mixed all our stuff up. We're slowly moving our way along, heading towards the colon. And here's the cool thing. There's this thing called a migrating mobility complex. And it's very frustrating that I can't move around the room right now. But what happens is, let's pretend you're a corn kernel. And so we come in through the stomach. Now, you know corn kernels, right? If you want to, you can notice that a corn kernel kind of looks the same on the way out as it did on the way in, right? Okay, so we've got a corn kernel. You are now a corn kernel. We're working you through. So what will happen is it comes through the stomach and it's still intact because, hey, you know, it's got a great cellulose coat, and so nothing is moving it into a breakdown product. So it's just staying intact as its little self. So it comes into the small intestine, and waves of mobility complex, in other words, peristalsis, will get it through and say we get down to here. And then the wave kind of moves along, but the kernel stays here. So let's say we're a corn kernel staying here and the wave kind of moves along. So then along comes another wave that starts down a little farther and it picks up the corn kernel. It's almost like riding the waves at the beach, where you get washed in a little bit and then the next wave comes along and then picks you up and moves you a little farther. Everybody with me on it? Okay, so those are called migrating mobility complexes. They're just this successive wave pattern that moves things more distally with each wave, okay? And it takes at least 2 of these to kind of move you from the beginning to the end. Because remember, we got a long way to go, right? Because it's 10 inches plus 8 feet plus 11 feet. That's about 20 feet of bowel to move through, just the small intestines. We haven't even gotten to the large intestine yet. So there's this thing called the gastro-ileal reflex. So you know gastro means what? Yes.
Stomach.
Stomach. And ileal is the ileum. So the gastro-ileal reflex is where when you put things into the stomach, you start ileal segmentation more. So in other words, we start stirring up this ileum of ours, getting it more active, and it relaxes the ileocecal valve, because when you get to the end of the ileum, there's a valve here to get into the cecum to start our trip through the large intestine tract, where we're really just compacting our feces. But right here, it's nice that it opens that door, because otherwise, right, we do our migrating mobility complex and all of a sudden we get down here and it goes and we can't get through. So the stomach, you put stuff in, it starts the whole stirring up down at that end. And remember, in the ileum is where we're reabsorbing our bile salts, okay? And you know what? It's gastrin who helps this happen. So remember, gastrin in the stomach, its big job was to make sure that we had enough acid and that we have enough of the pepsinogen to become pepsin. So it stimulated all that activity. Well, it's the one that goes down and says to the ileum, hey, you know, stuff's coming. You better get ready. Please open the door at the end so we can move along. All right. Good so far? Okay. I know those who did this already, you're already way ahead of this, so that's great. So now we're in the large intestine. So the large intestine's job really is to compact feces. Its biggest job is reabsorption of water. So we've reabsorbed most of it in the small intestines, but the large intestine is going to do the last of it. So we take this liquid slurry and turn it into something that's much more convenient in order to get rid of, okay? Keeps the brown trickle effect at bay. We have enteric bacteria, our normal gut flora, who are in charge of making sure that anything that is cellulose or something, they'll say, oh, I know what to do with that. And they turn that into methane, which means really literally we release methane. And there are people who have attempted to light flatulence. I did see somebody when I was at Vanderbilt, and yeah, he burned his drawers. So it worked, but he singed parts. But anyway, I have seen it work. So methane really is in your flatulence, and that you can thank your enteric bacteria for doing that. That's your next. Please don't do this. Okay. I mean, please don't do this. This is not a good idea. But anyway, the bacteria also are nice because they make vitamin K, and they make some of our B vitamins that we can get from our diet. So you don't have to have them, but it is kind of nice that we can make our own because we've got the bacteria doing it. You can totally live without your large intestine. Okay. You don't have to have it. There are a lot of people walking around with ileostomies. They are missing their entire colon. There are some people who chose the wrong parents, and they have a genetic defect where they are at such high risk for having colon cancer that they will actually prophylactically remove their entire colon, because you can't get colon cancer if you don't have a colon. So it's a pretty extreme thing to do, but when it's a liability like that, they will take it out sometime. Okay. All right. So on to the large.
Is there a genetic test
Yeah.
for that? Sorry. Is there a genetic test for that, like BRCA with the breast cancer gene? Is there something like that for colon cancer?
You know, I do believe there is. And but the biggest thing they do is history, because it's called familial adenomatous polyposis. So the best thing, the most important thing to do is, if it's in your family, if you have colon cancer in your family, is you need to make sure that you get a colonoscopy and they go in and look.
Okay.
Okay. But that's a great question.
But sorry, Sue, but
Uh-huh.
because my grandmother on my dad's side died from colon cancer. My dad had it. So I always have to do the colonoscopy. But they said because I'm too young, I should start it a little bit like when I'm 40, like in 10 years.
Start early screenings.
Yeah, you should start early screenings for absolute sure. I don't know. Do you have a gastroenterologist that you talk to? That you see?
Only when I was in Brazil, because I mean I moved here like 3 years ago.
Uh-huh.
But in Brazil they told me, yeah, you have a great chance, because if your grandmother passed away with colon cancer and your father had it and he had it removed, so you probably
Yeah, you're at high risk, hon. You need to, and I don't know, I would get a gastroenterologist in your life. I got a great one I like. So when you join with the team, you can email me and I'll talk to you about the guy I see, okay?
All right, thank you.
You betcha. Okay, so moving on, literally down the track. Okay, so we're going to move our way down through the large intestine. We know what we're mostly absorbing. We know about propulsion. In the colon, it's called haustral churning and mass movements. You got to love it. So the haustral churning is named because of these haustra. When you look at the large intestine, what has happened to the outer longitudinal layer is it's become these two thin ribbons that are called tinea coli, which means ribbons of the colon. So that's all that's left of the longitudinal layer. The inner circular muscle then, because of the way it's now constructed, has turned into these kind of like balloons. These are haustra, okay? So it's set up as a perfect tumble dry system. So imagine if we were together, I would be tossing a ball to the first person. Okay, so, and that would have actually, we probably would have started right up at the very front. I might have handed it off to Dominique. So, and she would have had to then bend with this great big thing. So imagine the haustrum catches it and gets really large as it goes. Oh, it's a big, heavy ball, right? So it's gotten this first bit from the cecum, from the ileum, and it goes, whoa, look what I've got. And then it contracts. It's a haustral contraction. So it's like taken up and then it dumps it to the next haustrum. So it slowly kind of tumble dries through here. Does that make sense? Can you visualize? So the ball is getting smaller and smaller as we suck it dry, working through. So it's like a tumble dry system. All right. So anyway, as we're moving along through here and we're getting drier, you need to know the path. So you need to go cecum to the ascending colon, to the right colic or hepatic flexure, transverse colon, left colic or a splenic flexure, down the descending colon, sigmoid colon, because it's shaped like an S, down to the rectum where we sit and wait until you say it's okay to leave. All right, so this is where you hold on to it, right? And then you've got your rectal valves holding everything in place, waiting to go down to the anus. All right, so all the way along, everything is absorbing more and more and more and more. Other little things hanging around here, just to point them out. The Vermiform Appendix, we met that because it's part of which system?
Lymphatic.
You got it. Tatiana, you are the bomb. All right, so it is part of the lymphatic system, actually, and I would imagine that Whitney could attest to this. You go mucking about in the abdominal cavity for some other reason. This is one of the things they'll kind of nick out of there because it's more of a liability than an asset at this point. It's what we call a vestigial organ. Remember that term? The VESTIGIAL, vestigial. You got it, but you don't need it, okay? And this one's kind of a liability. And then we've got these little yellow fingers of fat. These are the epiploic appendages. Again, your body says, you never know when the next meal might come, better store the fat. So these are little fingers of fat. We also like to hang out the fat on the transverse mesocolon and on that greater omentum. So you'll get fat deposition all over the place because we never know when the next spell of I'm hungry might come. All right. So anyway, when we get down into the rectum, so I'm going to move us down here. We'll zoom in on the rectum and the anus. So the rectum has the rectal valves. I mentioned this in lab a while ago now, but these are the ones that are the gift to us for social niceness. So if you have feces sitting in your rectum, then you can pass gas without passing feces. It makes things much tidier. You're more popular because even if you farted, it doesn't pass with other stuff too. So thank your rectal valves. Keep them happy. When we get down to the anal canal, this is when we start changing from, we go from the simple columnar and we go bang and turn into what? What would we have to change into when we're this close to the surface? What kind of epithelial tissue?
Non-keratinized, simple.
Not simple. You're on the right track though, Tatiana. You've got it.
Simple squamous.
No, stratified squamous. Stratified, remember, that's a whole bunch of layers, but you're right, it's non-keratinized and a stratified squamous. That's
like
because
lips, like our lips must
exactly
be.
Like our lips. Mm-hmm. So it's all about, because you know that corn kernel, remember we were all corn kernels going through, they're a little rough, especially if it's like popcorn kernels, because I don't know why you didn't break them up, but you evidently didn't enough. And so they're grinding down out through the back, and we've been drying out the feces as we've gone along. So anyway, now we also have internal and external anal sphincters. Which one do we control? Do you remember? Internal or external?
External. We control by external, and it's voluntary, and internal involuntary.
That's right. Okay, so external is stratified squamous. The internal are smooth muscle, or stratified squamous, stratified muscle, and the skeletal muscle, and the internal are the smooth muscles. So what happens is you get a load of stuff into the load of feces into the rectum, and it says, ooh, ooh, ooh. And this internal one is going to relax, and you hope that you still can control the external ones, because they're the ones who are going to tell you, okay, not now. If these give out on you, these external ones, that's when you have fecal incontinence, which nobody really wants because it's kind of a drag, right? Okay, so now down to, moving along, moving down. Let's see, where is my picture of, all right. So let's see, let's get to, there we go, to here. Okay, so I'm leaving this up for right now because it was on our train of thought, but I just want to make sure that you know that when we get down to this rear end part, the final part of our journey, the colon, when we look at it under the microscope, which we haven't done, but you have pictures of it in the lab to look at, but It has lots and lots of goblet cells again, because think about it, we're drying stuff out, right? So we need a sleeve of slime to move stuff along. So you're going to expect a whole bunch of goblet cells. But it's not as messy and chaotic anymore because it's doing minimal absorption. It's just water and some of the nutrients, the vitamins that the bacteria have made. Okay, so we don't have villi anymore. It's not got all that foldy stuff because it doesn't have to do as much in the world of the absorption. Okay, and so when we've moved, our haustral contractions have moved us all along and we've made it down through the sigmoid colon. We're sitting in the rectum now. Okay, so the journey is almost at its end, literally, figuratively, whatever. Okay, so what happens is you get a load in the rectum and there are stretch receptors that say, hey, we got something. And there are two things that happen. One is we have a spinal reflex that says, hey, something's in there. I'm good with that. Let's get rid of it. So then you have your parasympathetic nervous system, a spinal reflex comes down and says to the internal sphincters, relax, let's go. And we're going to talk to our levator ani muscles and we're going to start moving things up so that basically the feces that's sitting in here gets lower than where it was, simply because it's like, have you ever heard of that? Okay, I'm looking for a volunteer and nobody steps forward, but all but one person step backward. And now all of a sudden that person is forward. You heard of this? It's kind of like that with the feces. It doesn't really move anywhere, but the whole floor of your pelvis moves up. So there it is all of a sudden. And then the internal sphincters say, yeah, I'm good with that. But at the same time, we've got a message that goes up to the cerebral cortex that says, yeah, now's not good. You know, I'm in class and no. So you send the conscious control message down to your external sphincters and say, now's not good. So you pinch and you say, not now. And what happens then is that the stretch receptors die down. Okay, so now the involuntary reflex dies out. And what will happen is each time you get from your whole mass movements of the haustral churnings and moving things along, you're going to end up getting another load down into this rectum. Same thing's going to happen. This is the defecation reflex. It's saying, I've got stuff here, I need to move it. Here's the problem. We can hold on to this indefinitely, okay? And each time you get a load down in there, it's going to say, hey, the stretch receptors are saying, hey, let's go. But if you keep saying no, you can be literally anal retentive enough to put yourself into constipation, because simple columnar epithelial tissue is in the rectum and all the way up. So the longer stuff sits, the drier it gets. And you can end up with constipation, which is very uncomfortable. And then beyond that, if you really let it sit for a long time, and you get this with people who are on narcotics, which inhibits peristalsis. And so the feces sits, it doesn't move forward. And as it sits and dries out and dries out and dries out, you end up with something that's like wood putty. Then you need somebody who has at least an RN license to cover putting your finger up in there to dig out the wood putty. Okay. So the moral of this story is drink enough water and go to the bathroom when you need to. Okay. That's Sue's public health message of the GI tract. Everybody good? All right. So our mass movements, haustral contractions, we're out now. So for the clinical problems you are missing, and I don't even have it with me, but I have a really great model that has an incredible variety of problems on the GI tract. So we're going to go back to this picture. Whoops, I got to admit somebody. Chelsea just came, so let's admit Chelsea since she's just gotten here. All right. So, clinical problems. Hemorrhoids. Remember those? What are those? Besides unpleasant.
Isn't that, I think, like veins that have like the
Veins
valve?
that.
Yeah, they're not
Isn't it the varicose veins of the anus?
sluggish.
You got it. It's the varicose veins of the anus. Yeah, exactly. So varicose anal veins they can be internal or external. If they're internal, you may not see it, but you get bright red blood every time you go to the bathroom. And external are the ones that pop out and look really charming. And then people go get tucks and preparation H and stuff. The way to get those is a great way to get those is to let yourself get constipated a lot and bear down a lot and just put too much pressure down in there. Okay, so that's a great way. Another good way is to be pregnant, and then you got a big baby.
After pregnancy.
Yes, absolutely, because you got a great big kid pushing down and making it hard to get things out. Plus, you end up with constipation from pregnancy because progesterone inhibits peristalsis because it inhibits smooth muscle contraction. So you start pushing, and that's when you end up with the hemorrhoids. Okay, so we're good with hemorrhoids. Diverticuli. Here's the thing with diverticuli. Haustra are normal pockets, but when you get an abnormal extra outpocketing of your pockets, those are called diverticuli. Okay, so diverticuli are where we kind of are getting a weakening and an extra bubble blowout from the haustra. There are people who are prone to it. Sometimes they get little clusters of them. The problem with having diverticulosis, which is I have the pockets, is that you can end up with diverticulitis, which is inflammation of those pockets as things get stuck in there and you get an inflammatory process. And the thing that's scary about those is that the colon has what we truly would call feces. It's the bacteria-laden nastiness that nobody wants to have loose in their peritoneal cavity. So if you get diverticulitis with these pockets of infection and they rupture, you now have peritonitis and you can die of that. That becomes one of those, oh no, open you up, find where it's leaked out, flush you out, put you on some serious antibiotics, and then do a little praying because people can die of that, okay? It can be a real problem. So you got to be careful with that. They avoid things like nuts and things with little seeds that could get lodged in those. All right. And sometimes you have to actually have that piece of bowel taken out and put back together, resected. And that's when they'd probably go in and say, yeah, let's get rid of the appendix. What the hell, we're in there already. Okay. Diarrhea. Anybody not know what diarrhea is? Okay. That's your body saying, get it out real fast. Okay. So food poisoning will put you there, right, into diarrhea. It can give you nausea, it can give you vomiting. One of the great ones, kind of the classic food poisoning, is salmonella. That's the reason that you don't want to use your plastic thing to chop up your chicken and then turn around and do your veggie plate for your dip, right? Everybody knows that. Okay, that's a nasty one. Okay, so we know about constipation, we know about diarrhea, we're great. So now let's go back through the glands. So back up. All right, up we go. And we're gonna head up. We've already done the salivary glands, so I don't have to talk about those. Those were the first glands. Been there, done that, time for the liver. So in your books, you have to now go backwards. And I want you to go back up to here. So that is over on page 900. No, back up. I lied. Farther. 894 in your books. Okay. So we've seen the liver in the lab. You got the right lobe. You got the left lobe. You've got the, all of a sudden my brain just said, whatever. So we've got the major lobes. The right lobe is the biggest one of them, okay? Then if we look underneath it, we've got the caudate and quadrate adding into the left lobe. There's the falciform ligament running in between the parts. It's a connective tissue thing that helps hold it in place because it's the biggest gland in your body. It's massive. We got a little round ligament, ligamentum teres. It's a remnant from fetal circulation. We'll worry about that when we get fetal. All right. But the big thing about this guy that's sitting tucked up underneath your rib cage on the right side, it's huge and it does so many things. So This is my all hail the liver speech. Okay, so when you leave this course, I want everybody to think all hail the liver. It's amazing. It's also very pretty in its construct underneath. It's disgusting to eat. I don't think you should. It's disrespectful to the liver. So in your outlines, I have metabolic regulation, hematologic regulation, and biosynthesis. Okay, so these are all things done by the hepatocytes. You need to know about them. Okay, so the metabolic regulation. Remember, everything that goes into the liver from the small intestines, large intestines, stomach, and spleen. So all that stuff, all the nutrients that you've absorbed, all the toxins, if you have had wine with dinner, that stuff is going to head through the liver. The liver does the processing. So it's going to take nutrients, store some of them, let some through. It's not a perfect, I'm going to grab everything. It's going to take the toxins out of the blood. It has the ability to detoxify medications, alcohol, other drugs, whatever goes through the GI tract will go through the liver, okay? Becomes a really big deal when you're giving medications. If somebody has poor liver function, then we have to adjust the medications because it won't break them down as fast as we may need to. So we could put somebody into a toxic level if we have poor liver function and give them, well, this is how much we give somebody who looks like you. Well, no. If their liver's not good, then we can't do that. So it's a big deal to know about your liver function. It does absorb your fat-soluble vitamins, A, D, E, and K. You need to know those, okay? A, D, E, and K are DEKA, DECA, whatever. You need to know them, okay? They are fat-soluble vitamins. The liver will store them. If you are one of those people who says, well, if some is good, more is better, most is bestest, and you end up taking too much of any of the fat-soluble vitamins, you can put yourself into a hypervitaminosis state, which can be really, really bad. So you got to be careful, okay? So it's absorbing and storing those fat-soluble vitamins. When we look at what insulin and glucagon do, when glucagon goes looking for its energy stores, trying to raise blood nutrient levels, the first place it looks is the liver, because the liver's job is going to be to mobilize the stores. It's like our savings and loan bank for nutrients. The hepatocytes are going to break down the glycogen that they have created from the insulin, saying, well, you know, we got extra, let's store it. So it breaks down the excess nutrients, the glycogen that it's stored. It does lipolysis to break down the lipids. It's there that you can utilize amino acids through gluconeogenesis in order to create blood nutrients when you've run out of the glycogen, which is the ready cash in the underwear drawer or in your pocket, and then you've run out of your fats. We look at proteins, okay? So amino acids, the liver is the one who can do that magic of gluconeogenesis, all right? We've also looked at hepatocytes because we've met the renin, angiotensin, aldosterone system, haven't we? And remember, it makes angiotensinogen, which renin turns into angiotensin 1, which then we've got the ACE, angiotensin converting enzyme, turns into angiotensin 2, which does two things. Remember this one? Told you I'd beat you to death with it. Everybody good? Yes? Not dead? Okay. I've got these like deer in the headlights a little bit. I want to make sure that you're not dead yet. Okay. All right. So it synthesizes angiotensinogen. It's build the glycogen and lipid, stores fats when we got excess. It's the break them back down. So it's a savings and loan bank for nutrients. Thank your liver. Like, have you ever had one of those moments where you looked up and went, oh no, everybody else has stopped and I'm not ready to stop yet? And you jam on your brakes because, I don't know, you got distracted because you forgot to turn off your dumbification device, right? So you look up and you get that adrenaline surge, right? You can thank your liver that you don't spend the rest of your life going, adrenaline surge, it's the rest of my life. It breaks down hormones, it breaks down antibodies, it breaks down metabolic waste products, it inactivates our toxins. Your liver is amazing, okay? So everybody should really kind of look down at their lower parts of their body, look over on your right upper quadrant and say thank you. All right, hematological stuff. The liver stores a lot of blood. It's a hugely vascular thing. It's made up of, let me let Jacqueline in. So the liver is made up of these hepatocytes, and it's got all these sinusoidal capillaries, so it stores a whole lot of blood. Okay, that's why one of the dietary things you can do if you have an iron deficiency anemia, and you're willing to eat a liver, which, yeah, no, but if you are, it's a great source of iron because it's loaded with blood. So there you go. It has Kupffer cells, which are the macrophages of the liver. So remember, we like to name our macrophages based on where they are, if they hang out there for good. So the macrophages of the liver are Kupffer cells. You know how I feel about giving credit where credit is due. If somebody figured it out, I like to keep the name with it because I just think it's fair. So Kupffer cells, the macrophages in the liver, they're going to take out the bad red blood cells, right? So they break those down. They're going to deal with pathogens that come through the liver, because anything that came in through the junk, they clean up the junk. So they're great for there. We met the liver for hematological stuff. Remember the protein, the plasma protein? Do you remember what it was? Yes? No? Maybe so? Plasma. Protein. Big deal. Huge deal. Nobody? Don't remember albumin? Any ahas? Albumin?
In plasma, sorry.
Yeah, albumin. It's,
Albumin.
yeah.
Plasma protein. It's
Yeah.
albumin.
Right, albumin, the #1 plasma protein. But the liver also can synthesize cholesterol, and cholesterol gets a bad rap because if it's elevated too much, if you make it too well and it gets clogging arteries and stuff, that's a problem. But your hepatocytes are busy making albumin. They're making clotting factors, most of those procoagulants. In order to be able to stop bleeding, we need that for hemostasis, and it makes the cholesterol. So cholesterol is a big deal too. And a lot of what it's doing, it'll take the cholesterol, make cholesterol into the bile salts. So it's a big deal. It stores iron. It's a great organ. And that takes care of hematological. Then we've got the bile synthesis. And what is bile for, people? What's it do?
It is neutralize chloridic acid.
Emulsifies fat.
It does. It emulsifies fat. That's the right answer. Is it an enzyme? Yes or no?
Yes.
No.
No?
Thank you for playing. It is not an enzyme. It is a detergent. Okay, that's a big difference. The liver does not make digestive enzymes. It makes the bile for helping digestion by lipase, which is made by the pancreas, which we're about to get to right after this guy. Okay. All right. Moving along. Whoops. Let's see. So, didn't talk about those things. One moment, please. All right. So the other things in here, as I said, I'm just zipping when it's just anatomy stuff. We want to have time for the more complex things. So when you look at the liver, you've got your lobes, right and left. With the left has the caudate and quadrate, just leading into the left hepatic duct when we're talking about the bile and where it goes. Falciform ligament holding it in place. Round ligament we did used to be an umbilical vein from the fetus, but that's when we'll get back to that when we get to the fetus. The area where all the tubes are coming and going is the hilus, right? The porta hepatis is the hilus. We know that. That's an indentation where things come and go. We already knew that. All right, so the flow. You need to know the flow. We'll go with the flow. So we've got the two main lobes of the liver, the right and the left. So we've got right and left hepatic ducts, which lead to the common hepatic duct, which will then lead to the common bile duct. Now, the common bile duct, you get there and you have a choice. Do I want to go to the cystic duct and be stored in the gallbladder, or do I want to go down the common bile duct and join in with the pancreatic juice at the hepatopancreatic ampulla? So here's the thing, is that as you're flowing down this, okay, as the bile comes down, it comes down the cystic duct because we're closed off and not needed right now. When it goes into the gallbladder, in a moment we'll look at the gallbladder, but it's going to sit there. It's a muscular sac, but while it's sitting in there, it's going to get concentrated. We go from the really cheap store brand detergent, dishwashing detergent that you have to pour in like half the bottle when you've got a greasy frying pan in the sink, to the Ultra Dawn foaming dishwashing, one pump cleans the entire sink for you stuff. So it concentrates in the gallbladder. Okay, so that's my flow for that stuff. Those are the tubes there. Now, these guys, so in your book, turning the page, you see the way the liver is set up over on page 896, okay? So the liver is set up in these hexagonal lobules. Now, this is biology. They're not sharp-cornered things. They draw them as if they're kind of sharp-cornered, but they're not. But you need to know that it's set up with these liver lobules. We don't look like this picture up here. This is actually probably like from a pig, maybe a monkey. We are a little more less defined. We're a little less defined, more or less, whatever. A little less defined. We don't have these markings around us. But the construct is shown here. So what you'll see is that in each corner of these hepatic lobules, you have 3 tubes. So the 3 tubes are a branch of the hepatic portal vein. So it's bringing in the nutrient, maybe alcohol, whatever you've absorbed stuff from the GI tract, and including the stomach and the spleen. So when the spleen, the red blood cell graveyard sends its stuff over, all those things are going in through the hepatic portal vein, which then we're going in through the hepatic portal vein branch there. We've got a branch of the hepatic artery, the red one, because the hepatic portal vein's not bringing any of the oxygen. So we've got 2 blood supplies coming in, they mix. And then we've got this green tube, which is a little bile duct, which is going to be draining out. Okay, so following in let me get to a bigger picture of this one. Okay, so when we flow in, the blood flows in, it mixes. The hepatic artery branch, bringing oxygenated blood, and the hepatic portal vein branch, they join together and they flow through these sinusoidal capillaries. They're flowing past all these hepatocytes who are then doing all those things that the hepatocytes do. They're grabbing the nutrients, they're grabbing the toxins, they're detoxifying, they're building glycogen, building fat, or maybe we're breaking down, doing some glycogenolysis, we're lysing our glycogen, and lipolysis, lysing our fats, whatever. They're busy. They're very, very busy. And as we move along, slowly through the swamp of these sinusoidal capillaries, we're going to end up at the central vein of the lobule. The central veins then flow up and eventually join to hepatic veins. Remember when we went over the liver and had the hepatic portal vein, and I said, if you just leave out that portal, you have named the wrong thing? This is why. Because the hepatic portal vein brings in the nutrient-laden stuff, and the hepatic veins drain post-liver. So the hepatic portal veins will feed into the inferior vena cava, okay? So if I leave the portal off, I'm out after the liver. If I have the portal in the name, it's flowing into the liver. All right, so now the other piece that's happening is I'm making bile. So you know how in some older cities, and nothing in America is that old, but St. Pete or Seattle, you've got houses that have a street here, and then there's a street here, but there's a narrow alleyway between the backs of the houses. Y'all know what I'm talking about? Yeah. Where a garbage collection is and people go to park their cars. Okay, so the way your liver lobules are set up is you've got your hepatocytes, but running in between, there's like these back alleys. These are your bile ductules, okay? And bile canaliculi. Remember the bone had canaliculi? So you've got these bile canaliculi. In other words, little bitty biles channels, okay? So the bile canaliculi join together from these back alleys, from these hepatocytes, and they join to form these little bile ducts. So these 3 tubes at each corner are called the hepatic portal triads, or just portal triads, okay? So you need to know the 3 vessels that form those triads, because I will absolutely ask about that. So hepatic artery, hepatic portal vein, and the bile duct. Those are the portal triads. All right, everybody good? So flow in by blood, flow out with bile. Okay. So as we're making bile, there is a stimulus to make us secrete more bile, and it's related to the need. When you get chyme going into the duodenum, what happens is the duodenum says, we're going to need something to break down the fats. So you will get cholecystokinin, which I just like saying. It's one of the hormones the enteric cells make. So we've got cholecystokinin and secretin will be released when chyme gets into the duodenum, because those are the hormones that are going to then amp up the production of bile, the production of the pancreatic juices, and what else will happen is cholecystokinin is going to go back and it's going to talk to the gallbladder and say, we got food, it's in the duodenum, we need bile. Squeeze gallbladder. Cholecystokinin will get the gallbladder to contract. It's also going to get the sphincter of Oddi to open. It'll relax. So we're going to send the bile back up the cystic duct, because remember what came down there from the pathway, and then get to this common bile duct, and it's going to head out and squeeze out through this major duodenal papilla, because the sphincter of Oddi will relax. Okay? So, and at the same time, we haven't gotten to the pancreas yet, but it's going to cause the pancreas to amp up its production of pancreatic juice, which has all the different enzyme classifications we need, and bicarb, and they'll be released together into the duodenum. So we can thank our cholecystokinin for doing that. All right. And then remember, those bile salts are going to return to the liver because they're absorbed in the ileum. All right. I have some really cool pictures of a cholecystectomy, but phooey, you're there and I'm here. Becomes too much of a pain. All right. We good so far? Good sneeze, Thais. Good practice for being in public. All right. Let's see. So the, and by the way, the whole process of reabsorbing the bile in the ileum, we call it the enterohepatic circulation. So we put the bile in, we digest, we break down the fats into smaller globules, and then we get down to the ileum where we reabsorb it. Okay. Oops. All right, let's see. Where do I want to be? Not there. Sorry about that. Oops. Run away, run away. Okay. Let's get back to this guy. All right. So bile. Bile is made up of bile salts, which is based from cholesterol. And the pigment, and this is where it gets kind of, this is another cool factoid for when the conversation lulls in a gathering. This is your thing that you can like throw out and stop everybody from anything. And they'll think, oh my God. Why did you even say that? But here we go. So bilirubin is the primary bile pigment. You remember what bilirubin came from, right? What did it come from?
From erythrocytes.
Yeah, from the red blood cells. So it came from the heme group. So the bilirubin, which came from hemoglobin's heme group, it is the reason that feces is brown. It's the reason that urine is yellow. So in bile, we have this bilirubin that turns into, through a process, into something called urobilinogen, which gives the feces its brown color. So here's another interesting factoid. So if you have somebody who has liver failure, all the things that the liver does, now they're not working well. So they're bleeding, right? Their albumin is lousy. They have poor oncotic pressure. So therefore they're the puffy people because water leaves the blood vessels because it's not pulled in by the albumin's presence. And all sorts of other things happen. But the other thing is they can't deal with fats well. So when you can't deal with fats well, then because you don't have bile, because your hepatocytes aren't working, well, now what you have is if you eat any fat, the stuff that comes out the end of the pipe, the feces, it's called steatorrhea. I won't ask you about it, but anyway, it's this clay-colored, foamy, rotted fat stuff. It floats because fat floats in water, and it's clay-colored because it's missing the bile, the pigments, the bilirubin that would turn it brown. So it's this weird looking, stinky, rotten, fat floating, foamy stuff in the toilet. Maybe not a dinner conversation thing, but yeah, it's interesting. Okay, so clinical problems. Hepatitis. Hepatitis is not just the virus. Okay, you can end up with a variety of ways. It just means your liver's inflamed, but I'm going to just mention the two top oldest identified ones, hepatitis A and hepatitis B. Ready? So hepatitis A comes from fecal oral. Okay, so I'm going to give you Sue's memory aid that worked for me. And after I give you the B. So hepatitis B is blood and body fluids. So B for blood and body fluids. If it's wet and it's not yours, proceed with caution stuff. Hepatitis A comes from fecal oral. So fecal comes out of a place that has a slang term that starts with an A and then is followed by two S's. So for me, it was easy to remember that hepatitis A came from feces and hepatitis B came from blood and body fluids. So hepatitis.
I'm never going to forget that, by the way, ever. That's forever.
Never? See, and I said it without saying it. So there you go. I mean, if we're putting these on YouTube and somebody could stumble on this, I got to be careful, right? Okay, so anyway, hepatitis A or B, they're both pretty awful. Hepatitis A, you have to be careful. If you eat raw shellfish, please don't. They're filter feeders. And if you ever knew about the studies where they flushed a toilet and followed it with how fast does it show up in the water? Yeah, it's frighteningly fast. And so you eat that and you're basically eating other people's feces because it's filter feeders. So you got to be careful with raw stuff where people live, and we're everywhere now. So it's a little hard. Cook your shellfish, your filter feeders, please. Cirrhosis. If you have chronic inflammation of the liver, the next thing that'll happen is cirrhosis. Certainly hepatitis can put you there, whether it is viral or otherwise. Another good way to get there is to be an alcoholic. Chronic alcohol consumption will burn out your hepatocytes, basically. So the liver becomes inflamed and then it becomes fibrotic. And what happens then is if you think about the flow through the liver of all the blood, when you have cirrhosis, you can't get the blood through. Now it's kind of like you had a really big accident on the main road through town and now everything backs up. So as you start backing up, you get what we call portal hypertension. And remember, if I put a bunch of pressure on the blood vessels, I'm going to end up with fluid leaking out. So now you start getting these patients who, with portal hypertension, they end up with all this fluid in their bellies, in the peritoneal cavity, and they have to go in and tap it. And it looks like somebody just tapped a keg of beer and get that stuff back out. And when the liver isn't working, of course, all the other things, I mean, they have all these little micro bleeds, little bruises. They don't get cut if you have liver failure because they can't stop the bleeding well. They've got edema. They don't handle protein well. I mean, it goes on and on. But the cirrhosis, when it scars up, it means that not only am I not doing the job, but even the blood can't get through me and I get a backup. So that's called portal hypertension. Does that make sense? Can you kind of visualize that?
Yeah, I have a question.
Everything from the GI tract has to go through the liver. And if you block that down with scarring, you're in trouble. It's a backup. And then remember jaundice. What's that?
Ms. Shuman, before
Yeah.
you go any further, I have a question.
Yeah.
On the cirrhosis, I had a dear friend who was diabetic and got cirrhosis, and like you said, got the extended belly and went through the draining processes and whatnot. But they said she died mainly because of the ammonia that built up in her brain.
Yes.
How does that, the liver and the ammonia and all of that?
Okay, well, so one of the things the liver does, and we get to this in the very next chapter, but the liver in doing gluconeogenesis, when we take amino acids and we put them through the system in order to use them for energy, you have to clip off the amine group. Remember, it's amino acids. So there's an amine group, NH group. And then we've got the acid is the carboxyl group. So, and then you got the center carbon that has the whatever amino acid group it is. Okay. So, you know, if it has an H up there, it's a glycine. So you've got this carbon in the center of things, but on one side you've got nitrogen, which when we clip that off, then everything else that's left is carbon, hydrogen, and oxygen. We can use that to fuel ourselves, okay? Because we need carbon, hydrogen, oxygen in order to work with the mitochondria in order to generate ATPs. But that amine group, when you clip it off, it becomes ammonia. And your liver then takes ammonia and turns it into something called uric acid or urea. Okay, so the urea is non-toxic, and that goes out just as you would think in urine, right? That's the primary place it'll go out. It goes out in sweat and tears and all over the place, but urine, urea. So if your liver's not working and somebody has protein in their diet when their liver's not working, that ability to take that amine group and turn it from ammonia into urea is not working. So they get a buildup of ammonia. And, you know, if you've ever had a snoot of ammonia, just sniffed it, that's one of the things that they use to wake people up, is they put ammonia under their nose. And if you're faking being passed out, that will wake you right up. Okay.
Okay. Thank you.
You're welcome. All right.
Back to jaundice. Didn't mean to interrupt.
No problem. So what was jaundice? Is it a disease?
The skin's yellow.
Yeah, it's a sign, isn't it? It's a sign of liver failure, right? Was that what you were going to say, Tatiana?
I'm just going to say that it's very common in newborns.
Yes, it can be, because their little livers are not able to function well yet. So any red blood cells. Yeah, it's bilirubin. Absolutely. It's a bilirubin buildup, because remember, the liver is supposed to deal with bilirubin. And if it fails, then that's when you start getting the lack of bile production. It's when you start getting those clay-colored, floating, stinky steatorrhea poops, right? Okay. All righty. So gallbladder. Let me just mention back to that gallbladder. So I'm going back up to this picture right there. So I'm still hanging around the same places. But remember, the gallbladder, it really is kind of a green color. I'm going to guess that Whitney, at least, and probably Summer, but Whitney for sure has seen this. So they are really kind of a green shade because the bile kind of stains them. They're sitting down on the ventral side of the liver. They've got a real muscular wall, and its entire job, the gallbladder's job, is to store and concentrate bile up to 10x. Okay, so it does take it into a really much more concentrated, useful emulsifier, detergent. That's it. And it's the one that is connected to the whole pipe system by the cystic duct, which then goes backwards. The flow goes into the gallbladder and out of the gallbladder through the cystic duct. And then we end up going down the common bile duct again to the hepatopancreatic ampulla, where we mix with the main pancreatic duct contents of the pancreatic juice. And we go through our little sphincter of Oddi, which our cholecystokinin said, relax, we got to let stuff through. We got the gallbladder is contracting. Okay. So screw ups. When you look at the gallbladder, the big thing that happens is you build up gallstones. There's a fancy name for it. It's called cholelithiasis because bile is based on cholesterol. Bile salts are cholesterol based. So cholelithiasis are crystallized cholesterol in your gallbladder. Now, a lot of people walk around with this stuff and it doesn't matter, but if you end up with the gallbladder contracting and it gets a little lodged thing up in here, if you get a gallstone that lodges here, that's going to cause a whole lot of pain. A lot of people have some sludge in here. Another thing that can happen that really gets scary is if you get it up into, if it goes into, let me go, where am I? Over here. If it goes into right here, if instead of just staying in the cystic duct or the gallbladder, if it manages to get down the common bile duct and gets down to here, to where the hepatopancreatic ampulla is, then, sorry, I never get phone calls. All right. But anyway, then when it gets to this area right in here, if it plugs here, then it'll plug the flow through the pancreatic duct. And in the pancreatic juice, you've got all those digestive enzymes, including proteolytic enzymes, and it'll start to, digest the pancreas. So it can become very scary. Gallstones can be an extremely serious thing. You get this classic flank pain. It's back behind and it'll take you to your knees. All right. So it's something to not ignore. You know, you can say, oh, it's probably just a muscle strain. Well, careful. Okay. Because you don't want that to happen. And it can. It's not frequent, but it can. So mostly it's going to tell you it hurts, but that one's a bad one.
What's
All
the
right.
sludge when like a sludgy gallbladder? I've seen that on a lot of radiology reports.
Mm-hmm.
What is that?
It just means that you've got, it's like having sand in there. They're not big enough to do any damage. They're not going to plug anything up, but you've got this precipitate. It's like having sugar sand in there is really what it's like. I know, right? The worry is, are you going to get anything bigger? Be thinking, are they going to be like Legos?
Pre-indication to gallstones?
It could be a prelude, yeah, because you are precipitating your cholesterol, your bile salts. That's less than desirable. Okay, let's get the pancreas done, okay? So the pancreas is sitting there behind your stomach. It kind of looks like a tongue. It's got a head that's cupped by the duodenum. It's got a body and it's got a tail. The reason we care about head versus body versus tail really relates to if you get a screw up in there, it's a way to kind of state where the problem is, okay? So running along the length of it, this main pancreatic duct is just how we're going to carry most of the pancreatic juice. We've also got the little accessory duct. The accessory duct, or duct of Santorini, is actually just a dribble, kind of like dribble valve, you know, so it never, we've always got like a little low-level dribble of this bicarb juice stuff so that any chyme that comes dribbling through too will get neutralized. It's just this slow trickle. But when you want to bolus in because you've eaten something, that's when we're going to do the main pancreatic duct and go through that hepatopancreatic ampulla, through the sphincter vati, through the major duodenal papilla. Okay, so that's kind of the flow thing. So now, the pancreas, the way it's constructed, we met it with the islets of Langerhans, or pancreatic islets, making our hormones, right? But the bulk of your pancreas is actually made up of the acini, or acinar cells, and their job is to make the digestive enzymes, and they're going to make every flavor. They make an amylase, which is a carbohydrate, right? They make lipase, which is going to break down the fats, the lipids. That's the actual enzyme. They are going to make proteases, trypsin and chymotrypsin. Their job is to break down proteins. They make nucleases for RNA and DNA, okay? And then we also, in the pancreas, we make bicarb. So the bicarb is that base that's neutralizing the acid, okay? So when you look at the way the pancreas is set up, unlike the liver where everything is very organized, you've got your corners, you come flowing through from the portal triads, you work your way in, you got the central vein, out we go. If I played Honey, I Shrunk the Students, you could find your way out to the inferior vena cava. Here, if I said, get out of there, you'd go, oh my God, everywhere I turn, it's another dead end. So it forms all these cul-de-sacs. When you look at the pancreas, it's just this mess of individual cul-de-sacs. Okay, so these guys are busy making our pancreatic juice, and you definitely want to know that. Okay, now, when we look at what this pancreatic juice is comprised of, it's juice. So the number one ingredient obviously is what? It's
Water.
Juice. Water, of course, it's mostly water. Yes, indeed. So the amylase, the trypsinogen and chymotrypsinogen. Now, why would we have proteolytic enzymes that aren't secreted as trypsin and chymotrypsin? Why don't we just secrete them as they are?
Start digesting.
Yeah, it would be a real drag to digest yourself. That would be stupid. So trypsinogen and chymotrypsinogen work together. Okay, so trypsin then activates chymotrypsinogen into chymotrypsin. So these guys are things that are going to break down our proteins. Okay, so lipases, nucleases, and the bicarb is a really big deal because we've got to neutralize the stomach's chyme. Chyme is really acidic. Remember, the stomach is putting out a very acidic substance, and pancreatic juice is about a pH of 8 to try and counteract that nastiness. Okay? Everybody good? Okay, so far so good. All right, so the regulation of our pancreas doing its secretion is controlled by two things. One is parasympathetic nervous system. Excuse me, got a tickle. Anyway, so the vagus will stimulate the pancreas when you eat because we need it to be busy. But we also have secretin and chymotrypsin. Or chymotrypsin, excuse me, cholecystokinin. What was I thinking? That's like cholecystokinin is one of my favorite things to say. I can't believe I blew it. Okay, secretin. What a great name. What do you think it makes the pancreas do?
Secretes.
Secrete. Yeah, like duh, right? So secretin. This is secretin comes from the duodenum, and it is going to be secreted when hydrochloric acid gets to the duodenum and it says, oh my God, we need some bicarbonate. Holy moly, amp up the bicarbonate production because otherwise we'll get burned. We don't want duodenal ulcers. Then cholecystokinin from the duodenum is going to be secreted when you get the protein and the fats coming in, and it's going to say to the cholecystokinin, see, I get to say it a lot. Cholecystokinin talks to the pancreas and says, let's amp up the pancreatic juice secretion, and it's going to work with the secretin to make that happen. And remember, cholecystokinin is also talking to the liver. It says we need more bile, we need the gallbladder to contract, we need the sphincter vadi to relax, right? So you gotta love cholecystokinin. It's fun to say. Everybody okay so far with that? All right, we are almost done. Do you think you can stand the last little bit and then we're finished? Okay, because we're going to finish this chapter. That would be great. Okay. So the very last little bit is actually kind of table stuff. So if we go as in like not sitting on a table, but more like oh, for goodness sakes, that's too slow. Here we go. There. This is the table that is just wonderful. It's one of my favorites. If I were you, I would mark this in one moment, please, and I'll tell you where it is. It's on page what?
913.
Ooh, realize we're in the 900s. 913. Yes, this is a most marvelous thing. Know this one. This will be an important one to know the stuff on it, okay? All right, so let's look at carbs. You ready? Let's see what we've got here if we blow this up. So here are carbs. So you eat them, you got your starch, you got your disaccharides, salivary amylase, right? Breaks that up, pancreatic amylase. So the pancreatic amylase is released into the duodenum. So it works in the duodenum and down, works its way a little down into the jejunum. Then we've got our brush border enzymes. And remember, they're named by the disaccharides they work on. Lactose works on lactase, or is worked on by lactase. Maltose is worked on by maltase. Sucrose, sucrase. And we break them down into our constituent monomers, which are monosaccharides. These can then be absorbed. Everybody good? So lactose intolerant people are missing lactase. All right. Next, let's look at these guys. So proteins. When you bring in proteins, proteins are great big things. So pepsin starts it in the stomach. We start digestion of proteins in the stomach with pepsin because hydrochloric acid takes pepsinogen and turns it into the active pepsin. Because again, you can't have pepsin active when it's secreted, or you would start digesting the cells that make that. What were the cells that make pepsin?
Chief cells.
Yes, the chief cells, the ones where you're picturing an Indian chief chugging a Pepsi, because I'm so completely inappropriate. All right, so we're just breaking up our proteins, which are huge molecules, into smaller and smaller as we work along. So pancreatic enzymes, trypsin, chymotrypsin, carboxypeptidase, aminopeptidase. We get down to the brush borders until we get amino acids, which are our monomer units. When you see aminopeptidase and carboxypeptidase, it's just telling you which ends they're cleaving on. Because if I've got a chain of, like pearls, if you think of amino acids all lined up like a chain of pearls, that one end has the amine group on it and the other end has the carboxyl group. And so I can chop from this way, I can chop from this way until I've chopped them all off, okay? So that's what those mean. All right, and a dipeptidase, the old name for amino acids was peptides. So dipeptide, and they're peptide bonds that put them together. All right. All right, so anyway, so these, the brush border enzymes finish up the digestion in the small intestines. And then the very last thing are on our little color chart, are the nucleic acids. What happened to fats? Where did the fats go? Did I skip it? I must have. There they are. I was going to say, oh, all right. So, hi, Alexandra. We're just finishing up. So the fat digestion, we know there's a little bit of lingual lipase, but you know what? It's really just, it's a drag, honestly. It's why fat tastes good. It's why ice cream tastes good. It's why cheese tastes good, because you get a little taste there. It's not about really digesting them. It's to make you want fat, which I think is rude, but whatever. It's there. We've got a tiny bit in the stomach. Again, doesn't matter. But where we care is when we get down to the duodenum, where we've added in bile, which is not an enzyme. It's detergent and emulsifying. And we've got then the pancreatic lipase. So that breaks down the fats. Now, do you remember where they get absorbed? Where do fats go? Straight into blood?
No, into the chyle.
Yes, they go into chyle in the lacteals. These other things we've talked about, yeah, they go straight into the blood, which includes these guys. So nucleic acid, nothing happens to them until they get down to where the pancreatic nucleases work on them, and then we absorb those. Okay? All righty then. So when we're absorbing stuff, the big thing is not so much the details of active transport and all that, but there is a very interesting thing about the fat. So I'm going to kind of zip through like absorption. Most of the water and all the nutrients, really almost all of them, are absorbed in the small intestines. There's very little that's absorbed anywhere else. Most of them have to get through the cells, okay? Carbs use carrier molecules. There's a carrier molecule for glucose. Lipids, though, are the interesting ones. So I want to move down to the lipids, okay? So lipids do a cool thing, Okay, hi Heather, we're finishing up. We're almost done with this part. So the lipids, in order to absorb a lipid, so I want you to think of, water, vitamins, minerals, all that stuff, anything that's water soluble vitamins, so B's and C's, not the A, D, E, and K, right? They're going to come in, they've got carriers, active transport, some can just diffuse through like water can, but let's talk fat. So with fat, the cool thing is that when we break down the fat globule with our bile, because those are the bile salts, we've emulsified them. But in order to get into the simple columnar cells of this small intestine of ours, you have to form something that's called a micelle. So these are called micelles, M-I-C-E-L-L-E. So the micelles are fatty. And remember, they're trying to get through a phospholipid bilayer. So as they come through the phospholipid bilayer as a micelle, they get inside my cells. So in order to get inside my cell, I have to be a micelle. But once I get in there, now I'm in a watery place. So I had to be lipidy in order to get through the phospholipid bilayer of the simple columnar epithelial cells of this small intestine. Once I do, then I have to get coated with proteins. So I change my name into, and here's where, wait for it, chylomicrons. See, chylomicrons, micron for little. So as soon as I get into my cell as a micelle, I am coated with protein, which is compatible with the water. It's happy to be there, okay? Hydrophilic. And so now I'm a chylomicron. So fats are carried, lipids are carried as chylomicrons all the way through the lymphatic system, through the blood, okay? So got the difference? Because that's a big deal I'm going to ask. Okay, so my cells to get into my cell, chylomicrons from then on. All right, so that's how we get the fat soluble vitamins into. So A, D, E, and K hitch a ride through as a micelle and then are part of the chylomicrons. We've met B12 has its very own special thing to get in. What was that? Do you remember? How do we absorb B12?
Intrinsic factor.
Yes, exactly. The intrinsic factor. So intrinsic factor is how we absorb B12. And who made the intrinsic factor for the bonus realm?
Stomach cells.
Yes, the stomach. Do you remember which ones make the intrinsic factor? The ones that make two things. They make a pair of things. Parietal cells. You got it. That's it. Exactly. Okay, so the vitamin C and the B, most of the Bs other than the B12, they just get to passively diffuse through. But it's important to understand that the fat-soluble ones, A, D, E, and K, have to come in through micelles and become chylomicrons, okay? And they become part of the chyle, which is in the lymphatic system, all right? Other things come in, electrolytes come in through different ways. Most of them are some kind of an active transport. Iron, somebody has to carry them. When you look at the lab work, when somebody has labs done, they'll look at their iron carrying capacity, transferrin, transferritin. But anyway, so, and where is most of my water absorbed?
Colon.
In the large.
Nope, in the small intestine is most of it. We do the finishing of absorption in the large intestine. We good? Does that make sense? You realize that we've gone in the mouth and out the anus and done all the stuff in between. Woohoo! Okay, so now we're in the bonus realm of chat. And one of the things that I wanted to be able to do is for those who are doing the lab, I wanted to